students. Now I'm telling you about the other topics of the depression. Animal models for depression. This is for the PhD and MS students and also for the bachelors who do a job in the lab. Okay. Animal models for depression includes the number one is the open field test. Open field test is carried out before stress, 2 weeks after stress, and the 5 weeks after stress. The open field apparatus was a four-sided 80 cm into 80 into 40 cm wooden enclosure with floor painted white and divided into 25 equal squares by black lines and the side walls painted black. Tests are conducted in a dark, quiet room lit by light bulbs suspended above the center of the open field. Each animal is gently placed onto the middle square of the open field apparatus facing away from an observer and allowed to explore freely for 5 minutes. During the test time, the animal's movement in the field are quantified by counting the number of crossings, at least 3 paws in a square, and the number of rearings, wall rears, and free rear. Rear means to stand up. Stand up position of mice is called the rearing. Okay. After each animal is tested, the test apparatus is cleaned with a solution of 70% alcohol to eliminate odors for the next test. This is the animal model for depression of the open field test. Another one is the forced swimming test. The test is carried out on 2 successive days after the last rest period. Briefly, animals are forced to swim in a vertical plastic cylinder whose diameter is 21, height is 50, containing 25 centimeters of water, maintained at a 25 degrees centigrade. On the first day of experiment, animals are forced to swim for 15 minutes. On the following day, animals are re-exposed to the forced swimming for 5 minutes. During the second test session, the immobility time was evaluated by two observers who were blind to the kind of treatment each animal. An animal was judged to be immobile whenever it remained floating passively in the water in a slightly hunched but upright position with its head just above the surface. At the conclusion of the swim test, the animal was removed from the cylinder, dried by a towel, and returned to its home cage. The water was changed after each rat swimming. This is apparatus for the forced swim test. Here is the immobility of mice, there is a swimming, and it's the climbing. Animal model for depression includes a sucrose consumption test. Animals are trained to consume 1% sucrose solution before the experiment. They are exposed to two bottles of 1% sucrose solution in a 24-hour period and exposed to one bottle of 1% sucrose solution and one bottle of drinking water for the next 24-hour period. Formal test is carried out before stress, 2 weeks after stress, and the 5 weeks after stress. Animals are kept food and water derived for 21 hours and they are exposed to both the test solution. 1% sucrose in drinking water for the next 1 hour period. Sucrose consumption was measured by re-weighing pre-weighed bottles of test solution. Bottles were counterbalanced across left and right sides of the cages throughout the experiment, varying for each test and each animal. Another one is a tail suspension test. Primarily used for mice, the animal is suspended from the tail and the time to an immobile posture recorded. Mostly not done under for the rat because rat weight is heavy and only this experiment is mainly used for the mice. Number fifth is the olfactory bulbectomy. The animal is placed in the stereotaxic apparatus and the skin incision is made on the skull. A hole is drilled into the skull to access the olfactory bulbs. The bases of the bulbs are separated and the olfactory bulbs are aspirated. The skin incision is closed with wound clips or per request. Bilateral destruction of the olfactory bulb in rat is considered a validated model of major depression. The surgical procedure leads to a wide variety of behavioral, neuroanatomical, and the neurochemical changes. Bulbectomized rats should reduce levels of noradrenaline and serotonin, And these neurochemical changes provide construct validity and particular support for classification of the procedure as a model of depression.